The inner ear, that is also called as labyrinth, is made up of two parts, the bony labyrinth and membranous labyrinth. The bony labyrinth is a series of channels in petrous portion of temporal bone. The membranous labyrinth is present inside these channels and it is surrounded by fluid called perilymph. This membranous labyrinth more or less duplicates the shape of the bony channels and it is filled with a fluid called as endolymph. There is no communication between the spaces filled with endolymph and those filled with perilymph. So this diagram shows the middle ear and the inner ear. When the sound waves falls on the tympanic membrane, the ossicles, they vibrate and the vibration is transmitted to the inner ear through the uh, to the cochlea, and cochlea has the three parts: scala vestibuli, scala media, and scala tympani. Scala vestibuli continues with the scala tympani at the helicotrema. So whenever sound waves falls on the oval window, the sound waves produces vibration in the scala vestibuli, so which comes back through scala tympani. Okay, so vibration of these scala vestibuli, it also leads to vibration of the scala media, so which harbors the organ of corti. Within there'll be hair cells. The distortion of the hair cells is going to produce the action potential which reaches the brain, and the sound is sensed by the brain in this way. The internal ear has divided into three chambers, that is the scala vestibuli, scala media, and scala tympani by two membranes, that is resinous membrane and bacillus membrane. Scala vestibuli and scala tympani are filled with perilymph and are continuous at the apex, that is through the helicotrema. Scala media is continuous with membranous labyrinth and it is filled with endolymph. Here you can see the cross section of the cochlea, so which through the two membranes, that is basilar membrane and resinous membrane, divides the canal into the three parts: scala vestibuli, scala media, and scala tympani. Scala vestibuli and scala tympani, they are continuous and they have in them the perilymph, whereas scala media, it is filled with the endolymph. And here you can see the organ of corti, so which is a sense organ of audition, which harbors many hair cells in them. Organ of corti. It is located on the basilar membrane and it contains hair cells. Hair cells are the receptor cells that transduce sound energy into electrical energy. It extends from the apex of the base of the cochlea and has a spiral shape. There is one organ of corti in each ear. The process of hair cells, the PS, the tough membrane like reticular lamina that is supported by rods of corti. The hair cells are arranged in 4 rows and 3 rows of outer hair cells lateral to the tunnel formed by the rods of corti and 1 row of inner hair cells medial to the tunnel. There are around 3500 inner hair cells which are important for fine sensory transduction, and there are about 20,000 outer hair cells which is responsible for detecting the presence of sound. Outer hair cells are capped with gelatinous material called as tectorial membrane. Cell bodies of afferent neurons that surround the bases of hair cells are located in the spiral ganglion. About 90 to 95% of fibers innervate inner cells and only 5 to 10% innervate outer hair cells. In each hair cells, there are about 30 to 50 hairs called stercocelia, and it has actin and myosin. Very fine processes called tip links tie the tip of each stercocelia to the side of the its higher neighbor. At the junction, there are cation channels which are mechanically sensitive. When hair cells move in one direction towards the longer stercocelia, channels open up and there is the influx of potassium and the calcium. If the cells move in opposite direction, the ion channel closes. The supporting cells are in inner phalangeal cells and outer phalangeal cells. Inner phalangeal cells, they support the inner hair cells, and outer phalangeal, they are also called as diater cells, support the outer hair cells. The outer phalangeal cells are supported by Hansen cells, which again supported by the Claudius cells. The junctions between hair cells and other supporting cells are tight junctions to prevent mixing of the endolymph and perilymph. So this is the structure of organ of corti, so which is based on the basilar membrane, which is, and it is made up of a tunnel and medial to which we have the inner hair cells and lateral to which we have outer hair cells. Inner hair cells are less numerous compared to the outer hair cells, and the tips of the hair cells, they are embedded in the tectorial membrane, mainly that of the outer hair cells. And you can see the nerves which forms the spiral ganglion from hair cells, so which helps in the transmission of the action potential from these hair cells. And the other supporting cells for the outer hair cells are cells of Claudius. Sound transduction. Sound waves which enter the cochlea gets transmitted along the scala vestibuli and spreads towards helicotrema. At the same time, sound waves are also transmitted to the fluid into the chambers, into other chambers. This will lead to the vertical up and down movement of basilar membrane. This results in up and down and side to side movement of hair cells. The process of hair cells project into the endolymph, whereas bases are bathed in perilymph. The mechanism of production of action potential. When tip links pulls the stercocelia, the ion channel opens and there is a potassium influx because the endolymph consists of more amount of potassium. The receptor potential occurs in the hair cells, which causes the calcium influx, which causes the depolarization of the hair cells in the lower part of the hair cells, and that causes the release of neurotransmitter, that is glutamate, into the synaptic cleft between receptor cell and afferent nerve, sensory nerve. And neurotransmitter eventually binds with a specific receptor on the sensory nerve fibers, and ligand gated ion channels open, which causes action potential in the cochlear division of the 8th cranial nerve. And then the